right, so mitosis and cytokinesis produce two genetically identical daughter cells. So when cells in my skin are dividing, when one cell divides into two, those cells here are genetically identical. So how does that happen? So now we're going to take a look at that and look at some of the phases of a dividing cell. So interphase, if you remember back in that cell cycle, interphase is broken up into G1, the gap 1, the S, and the G2, and that's called interphase. It's in between cell divisions. And then the DNA gets ready to go, and we wind up with a cell that kind of looks like this. So I'm going to show you some pictures as well as some illustrations. So within the nucleus, we have the DNA that's just doing what it's doing. And then we also have these structures called centrioles that are going to produce kind of the railroad tracks that are going to drive the chromosomes around in cell division. So mitosis is broken up into 4 phases. And the first one is called prophase. Okay, so what happens in prophase? Write this down. Here we go. Chromosomes condense. So in other words, the DNA that's all spread out and being used by the cell condenses into these structures that look like the chromosomes you have here. Now, how many chromosomes are there in the cell? Count them. I can't hear you. There's 4. Yeah, there's four chromosomes, and they're going to show up in prophase. So prophase, next step is metaphase. And metaphase, what happens is that the chromosomes line up in the middle of the cell. So they've been pushed and pulled by those lines. Really cool motors is what they are. There's some, it's really, I'll show you some animation of that at some point in time. And actually some live footage, because you can watch these guys on those spindle fibers doing what they do. So the metaphase cell has the chromosomes lined up in the center, on the equatorial plane, as it were. In the middle, there's that illustration, but then over on the left side of your screen, you'll see the spindle fibers have been stained green by a fluorescent dye, and then the chromosomes are stained, of course, I don't know because I'm colorblind. It's either blue or purple, I can't tell. And so that's an actual cell that's going through division. So we've got prophase, we've got metaphase, now we've got anaphase. Now the sister chromatids are going to separate, and they're going to get pulled by the spindle fibers, by these little walking motor things, to the poles, to the opposite sides of the cell. So you can see the red chromosomes getting pulled in the photograph, as well as the 4 chromosomes that are getting separated in the illustration. So that's anaphase, prophase, metaphase, anaphase. And the last thing is telophase. And we start to form 2 nuclei, and the cell starts to split apart. Notice the chromosomes are different than in the beginning. They're not replicated anymore, but there are still 4 chromosomes in each of these cells. So remember, the goal of mitosis is to produce 2 genetically identical cells. And so that's kind of what's happened. Now, cytokinesis is the splitting of the cells. And it's a little bit different in plant and animal cells. So we'll start with an animal cell. Here's an animal cell that has 2 nuclei and some proteins called actin filaments are going to pinch this cell off. It's going to pinch it off and just create a crease between these 2 cells until eventually we're going to have 2 brand spanking new cells. So that's what the cells in the human body do. That's what the cells in any animal do. So they actually have this thing called a cleavage furrow, where the actin fibers kind of, it's like a purse string, if you will. Now, in plant cells, we form a cell plate. So here's a giant plant cell that's undergone cell division. And remember, plant cells have cell walls. And so it's going to form a cell plate that's eventually going to form a new cell wall made of cellulose. So a little bit different for plant and animal cells. Differences there. Otherwise, not much different. They're doing, holy cow, look at this. This is a picture of an onion root tip. And look at all these different cells, and there are different parts. Can you find prophase? Can you find an anaphase cell or metaphase? you should be able to do that. And that's one of the things you're going to be working towards. So prophase, metaphase, anaphase, and telophase. So mitosis and cytokinesis. So here's a look at cells in interphase. This is live footage of cells. It's sped up a little bit. But you're going to watch these cells go through cell division. Ooh, prophase, look at that. You can see the chromosomes starting to show up. The nuclear membrane goes away. It's kind of like I'm doing play-by-play. As the chromosomes condense, now we're just about into metaphase, Jim. That's right. They're lining up in the middle of the cell. There we've got lined up nicely on the metaphase plate. The centromeres get pushed and pulled to the center of the cell. Anaphase, there they go. Look at those chromatids. They've been separated now, and now we have telophase cell. Way to go, Jim. Way to oh, look at that. And we've got 2 new cells. This is a plant cell, so eventually that line is going to form 2 brand spanking new cells. And that's cytokinesis. So mitosis and cytokinesis going on in those cells. Okay, just a quick review. Think about the chromosome anatomy. That's going to be really important. Chromosomes, chromatids, centromeres, sister chromatids, all that stuff. Interphase and G1 and S and G2 and what happens in those. So what happens in those, that'll be an important thing to know in the cell cycle. And then mitosis and cytokinesis, what happens in prophase, what happens to the chromosomes, what happens in metaphase, what happens to the chromosomes, what happens in anaphase, think about the chromosomes there if you have to go back and look at it. And then telophase, and we're going to make eventually 2 nuclei, and then eventually pinch those off into brand spanking new cells. So mitosis and cytokinesis, the business of cell division, making 2 genetically identical cells. So I hope that helps.